Okay, I guess we'll get started. Hello, everyone. Welcome to the ARO seminar series. The title of today's talk is Regeneration of Hair Cells After Damage, a Historical Perspective and an Update on Mammals. My name is Mark Warshall. I'm a professor in the Department of Otolaryngology at Washington University in St. Louis, and I will be moderating today's session. Before I introduce the speaker, let's just go through a few organizational details. I'd like to draw your attention to the tools located at the bottom of your screen. In order to preserve the flow of this talk, we will not answer questions until the end of the talk, and you can state questions using the Q&A function that whose icon you see down in the lower stream. You can post questions anytime during the talk, and they will be read by me and then answered by the speaker at the end of the talk. When you do post your questions, be sure to include your first and last names so that the speaker can get back to you if there's not time to answer the questions. And I should also point out that your name will not appear in the Q&A dialog box. So there's closed captioning that's available for this webinar, and it's being recorded. We also hope that you will participate in the post-webinar evaluation so we can get feedback and hopefully improve these as we go along. And then finally, if you're experiencing any technical difficulties with receiving the talk, you can send an email to headquarters@aro.org, and there'll be somebody there to see your query and hopefully resolve the issue. So with that, then I'd like to introduce the speaker of today's talk, Dr. Jennifer Stone. Dr. Stone is a professor in the Department of Otolaryngology at the University of Washington in Seattle. Her research for most of her career has focused on mechanisms of hair cell regeneration in the ears of birds and later in mammals. She did her graduate work at Boston University with Doug Kotanch, who was one of the pioneers in studying hair cell regeneration in the avian inner ear. Went on to do a postdoc at the University of Washington with Ed Rubel, who's another pioneer in that field, and then has remained at University of Washington ever since. People who move to Seattle sometimes are reluctant to leave, I guess. But she has been working on regeneration in the avian inner ear and has really pioneered a lot of really nice work focused on regeneration of the vestibular organs of mammals. And I expect we will be hearing about both of those in the talk. So without any further word, I'd like to give the screen over to Dr. Stone.
Hello, Mark, and thank you, Mark. Appreciate it. I want to start by thanking Sparrow for the invitation to speak today, and also recognizing the efforts of Ali Jevons and with Parthenon for organizing this wonderful series. It's really an honor to be asked to speak today. ARO has meant a lot to me throughout my career. I attended the first time as Doug Kotanch's graduate student, and that very year I met Ed Rubel, and ever since then, I have been really relishing attending ARO every year, seeing old friends, meeting new ones, learning about research in our community. And I'm going to sincerely miss that this year. So I'm happy to see you this way. So today I will provide an overview of regeneration. And I'll start with a historical overview, primarily talking about hair cell regeneration in birds, where I've done most of my work. Talk about the discovery of this process, the cellular mechanisms that occur, and some of the molecular regulation that controls hair cell regeneration. And then I'll provide an update on mammalian hair cell regeneration. And most of this will focus on the vestibular system because that's where most of my work's done. But I'll try to also address some of the current thinking in the auditory regeneration research in mammals. I have a couple disclaimers, which is this is a very big field and it's hard to provide a big overview without excluding things. And so I'm only going to talk about two species, primarily birds and mice today. And I acknowledge the work of all the people whose research I can't talk about. So hair cells are mechanoreceptors that sense the energy in sound waves and in head motions. And they're shown in these beautiful images here from Jim Hudspeth and David Corey. The primary sensory organelle in these cells is called the hair bundle, and it's shown here where multiple stereocilia are located, as well as a single kinocilium. Hair cells are the only cells in the body that are able to do this mechanotransduction and to sense energy and sound waves and head motions. And therefore, when we lose them, we have profound sensory deficits. So I want to talk a bit about what the rationales for studying hair cell regeneration are. 15% of adults have hearing loss, and about 35% of adults over the age of 40 have balance deficits. So auditory and vestibular hair cells have they die as a result of several things, including mutations, acoustic overstimulation, ototoxic agents, infections, and the aging process. And it's important to acknowledge that not all cases of hearing loss or balance dysfunction are due to hair cell loss. Synaptopathy and neuronitis cause hearing loss, as do dislodged otoconia, and vascular stroke can cause vestibular dysfunction. However, loss of hair cells often results in irreversible sensory and sensory motor deficits, including deafness, tinnitus, dizziness, loss of balance, and oscillopsia. So it's possible that hair cell regeneration could result in better restoration of hearing and balance function than the current treatment options. And it's also possible that hair cell regeneration could augment current treatment options, such as cochlear implants. Now, all of us, if you're not already doing so, will experience age-associated loss of hair cells in hearing and balance organs. And this is illustrated here for the cochlea and for the utricle, and work from Wu et al. And Taylor et al. From the humans that really had no known history of noise or drug damage, and yet show considerable loss of hair cells in the cochlea at a variety of ages, and also in the utricle. In this particular utricle from an 80-year-old female, there's only one single bundle left in the field, and here, 65-year-old male, we see no bundles. And this guinea pig image here, scanning electron micrograph, is just shown to give you an idea of what the normal density of hair cells should be. So hair cell loss is a significant problem, particularly as we age. And the good news is that all of these non-mammalian vertebrates naturally regenerate their hair cells. And they provide us with a model to study, or several models to study. Work in the early 1980s by Jeff Corwin and Art Popper looked at normal addition of hair cells in fish and amphibians as part of the post-embryonic growth period. It wasn't until the early or mid 1980s that it was discovered that birds, which are warm-blooded vertebrates, can regenerate their hair cells after damage. And this work in birds sparked a flurry of other studies in these other non-mammalian vertebrate classes. So the avian hearing organ is what I'll focus most of the talk on, or the first half of the talk on anyway. And it's shown here. The hearing organ is located in the cochlear duct, which projects ventrally in the avian inner ear. And the organ itself is sort of a flat, elongated, sickle cell shaped structure that's tonotopically organized like the cochlea. And when we look at the sensory epithelium, which is also called the basilar papilla or the BP, we can see the individual hair cell bundles projecting up and the hair cells each surrounded by a non-sensory or 2 to 3 non-sensory supporting cells, which kind of look like a necklace here. We look at a cross-section of the basilar papilla, we can see its structure further with the hair cells located along the scala media and the supporting cells interdigitating between them and extending down toward the basal lamina here. The supporting cells are actually quite long and thin cells. So studying tonotopy in the BP, Doug Kotanch was using pure tone loud stimuli when he accidentally discovered hair cell regeneration. This is how hair cell regeneration in birds happened. It was discovered, it was quite by accident. And Doug had been giving chickens loud, intense noise at particular frequencies and then looking at the BP afterwards. And these noise episodes cause large swaths of hair cell loss and supporting cells expanding in that region. And when Doug waited over a week and looked at his samples, he was delighted to see what looked like very immature baby hair cells that had grown in that region. And because Doug had worked on development, he recognized that these were immature hair cells and probably represented regeneration. And Ed Rubel's group was studying ototoxicity in the BP as well. And he found that numbers of hair cells increased after damage that was caused by the ototoxin gentamicin. So here's the main hair cell density on the y-axis and the percentage basilar papilla length on the x-axis. And what Ed, and you can see the control hair cell numbers in the black line here across the length of the BP. And when they gave gentamicin, Raul Cruz did so, there was a significant loss of hair cells throughout the high frequency end of the BP, but over time, 22 days, these numbers increased, and Ed reasoned this may be regeneration as well. The definitive evidence for hair cell regeneration in birds came from work from Corwin and Kotanch and Riles and Rubel in the late 80s. And what they did is they gave multiple injections of tritiated thymidine, which marks dividing cells and their progeny, to birds after noise damage. And then they looked at about 10 days after noise damage. And what they found was that tritiated thymidine, which creates these sort of grains, audioradiographic grains over cells that have divided or are dividing, labeled hair cells as well as supporting cells. And this indicated that these cells had been generated anew and were therefore regenerated. So the structure of the basilar papilla is restored during hair cell regeneration, quite remarkably. These are scanning electron micrographs showing a normal BP, a noise damaged BP, and a regenerated BP at about 30 days after damage. And what's important to state here is that the vestibular and auditory hair cells are regenerated regardless of the type of trauma they undergo, whether it's noise damage or ototoxins or laser ablation. And all hair cell subtypes in birds appear to be regenerated, the tall and short hair cells in the auditory system, and the type 1 and type 2 hair cells in the vestibular system. But perhaps most impressively, hair cell regeneration restores function. This is shown really beautifully in this study from Riles and Dooling groups, where they demonstrated that budgerigars or parakeets, after regeneration has happened, are able to recognize calls that they learned before damage had happened. So they trained these birds through operant conditioning to learn certain bird calls, which are complex auditory stimuli. And then they tested them to see if they recognized the call. At that point, they push a button, or if it's a call they were not trained on and didn't recognize it, they just went on to the next trial. And these birds were actually quite talented, and they learned how to do this task very well at about 90% efficiency, or percent correct. But after kanamycin, the ototoxin was administered and hair cells were lost, their likelihood of getting the answer correct dropped to 50%, which is about chance. But after about a month, as the hair cells were regenerated, these birds regained the ability to recognize the calls with great efficiency. And so this is a beautiful representation of the restoration of auditory function in birds. So of course, people were interested in how new auditory hair cells are regenerated in non-mammalian vertebrates. I think people were really surprised to see regeneration, particularly in warm-blooded animals. And so some of the studies in the early 90s addressed this question, and they did it through a variety of approaches, but I'll show one example here, which is again using tritiated thymidine. They did a pulse fix paradigm where they gave tritiated thymidine to birds after damage and then waited a short period of time. And what that does is it allows tritiated thymidine, which is incorporated into DNA synthesis, it allows it to be incorporated in DNA synthesis, but then when they look at the birds right away, it allows them to catch cells while they're still in S phase. And that identifies the progenitor cells. So in the undamaged basilar papilla, we don't see any grains of tritiated thymidine over any cells because there's no production of cells in normal conditions. But after damage, 3 days after gentamicin, we saw uptake of tritiated thymidine in cells that are remaining after damage. You can see the epithelium shrinks a lot as it's damaged. And these cells are supporting cells. And so this was the evidence demonstrating that supporting cells are the progenitors to new hair cells. But interestingly, some new hair cells in the BPs are not formed by supporting cell division. And this was demonstrated again by several groups around the same time. And I'm showing data here from our lab on cultured BPs. So we damaged cultured BPs with streptomycin. And then we provided the BRDU, which is a marker similar to tritiated thymidine that marks dividing cells in their progeny. We provided BRDU for a long time in culture. And then we looked afterwards. And as we expected, some of the new hair cells, which are labeled with antibodies to myosin 6 in red here, had the BRDU. But we were surprised to see that some hair cells didn't have BRDU, indicating that they didn't arise via cell division because this marker was present the whole time, and they would have done so. They would have had BRDU in them if they were post-mitotic. So then we hypothesized that if we blocked cell division here, we should still get these cells if they don't rely on regeneration. And so we did that with a cell division marker called acetylcholine, and indeed hair cells were regenerated nonetheless. So regeneration in the BP happens in 2 ways. It happens through supporting cell mitosis, which is shown here, where after damage, hair cells are ejected and supporting cells re-enter the cell cycle and divide, and their daughter cells go on to differentiate into either hair cells or supporting cells. And then they mature, and within a month, this epithelium looks pretty normal and function is restored. Regeneration also occurs through this non-mitotic mechanism I mentioned, which we call direct transdifferentiation. And in this process, after damage, supporting cells phenotypically convert into hair cells without dividing. They downregulate their own genes and phenotype, and then they acquire the hair cell phenotype and mature. It's important to acknowledge that both mitotic and non-mitotic regeneration occur at the same time, making new hair cells, and that mitotic regeneration probably is a major contributor to replacement of the supporting cell population. So which molecules regulate hair cell regeneration in non-mammals? Well, I'll talk a little bit about this in the bird. There are a lot of studies on this, but I'll highlight a few. So ATO1 is a transcription factor that has been studied extensively in hair cell regeneration. It was demonstrated to be a developmentally critical transcription factor because if we knock out ATO1, if people do that in mice or other species, hair cells don't form. Or if ATO1 is overexpressed or misexpressed in an area where it shouldn't be, then hair cell hair cells are induced there. So ATO1 is both necessary and sufficient for hair cell differentiation or during development. In a mature basilar papilla, because there's no ongoing hair cell production, we don't see any expression of ATO1 when there are a lot of hair cells present. But right after damage, 3 days post-genomycin, we see upregulation of ATO1 in almost all the supporting cells in the area of damage. And this is protein here. As new hair cells differentiate, what we see is a shift in the ATO1 pattern where it's downregulated in some supporting cells and upregulated in others, so that a lot of the cells have a high level of ATO1 expression. And when we look at the fate of those cells, we find that those cells are differentiating into hair cells, as we might expect. So they're expressing myosin 6 and ATO1. But once hair cells have been fully restored, then ATO1 is downregulated again. So if we look at the stages of hair cell regeneration in the chick BP using ATO1 as a sort of marker, we see in the normal epithelium with lots of hair cells that supporting cells are quiescent. They do not express ATO1. After hair cell damage, they leave quiescence and upregulate the gene. And then what happens is this process called hair cell precursor selection, during which some cells downregulate ATO1 and some upregulate it. And those cells go on to differentiate into hair cells. The cells that downregulate it differentiate into supporting cells. And then after this is all done, the pattern returns back to before damage. So based on what I told you about ATO's role in development, you are probably wondering whether overexpression of ATO in the chick BP can cause more supporting cells to turn into hair cells. And that is indeed what happens. In the study from Becky Lewis, she cultured BPs and then transfected them with various plasmids, transfected supporting cells with various plasmids, and then looked at either 2 days or 7 days after the transfection. And at 2 days, this is what these labeled supporting cells look like. They're elongated cells throughout the BP. And after the later survival time, some of these cells differentiated into hair cells, as shown here by this green cell with a myosin 6 labeling. And some remained as supporting cells, as we would predict. In a negative control experiment, she found out that about 5% of the supporting cells that were electroporated acquired the hair cell fate. But when ATO1 was overexpressed by the gene she transfected, this was about 70%. So ATO1 overexpression caused a large proportion of the cells that were going to become supporting cells to become hair cells. Similarly, she studied another pathway, Notch signaling. And Notch, in a simplistic description, inhibits transcription of ATO1 and accumulation of ATO1 protein. So she reasoned that inhibition of Notch signaling should allow more hair cells to differentiate as well. So in this case, she transfected the control plasmid that allowed her to visualize the supporting cells, and then provided the Notch inhibitor, DAPT, for the rest of the experiment. And what she found was in her negative controls, there was a greater degree of differentiation, probably because they had this element, this drug called DMSO in there as well, which is a vehicle control for Notch inhibitor. But with Notch inhibition, there was a significant upregulation of the new hair cells that were made. So to sort of summarize what we think ATO1 and Notch do in regulating regeneration is we believe that ATO1 drives this hair cell precursor selection and differentiation, and that both of these processes are modulated by Notch. We don't think that Notch inhibition is what leads to supporting cells originally leaving quiescence in the chicken. So I'm going to finish up this section on non-mammalian regeneration and just mentioned that vestibular hair cell regeneration also occurs in birds. And it's known to occur in the utricle and the crista. And it happens after ototoxic drug treatments using kind of the same mechanisms that the BP uses that I just described. And some of these studies make good reading if you want to read more about that. All of them make good reading. And then it's important to also say that vestibular function, as measured by the vestibular ocular reflex and the vestibular colic reflex or motor behaviors, is also restored, but it takes longer than the BP, several months. And these studies are really great studies to look at to understand more about that. So now I'm going to switch over to talking about mammalian hair cell regeneration. And we've known for a long time that hair cells are not replaced in the cochlea of adult mammals. And these are images from Andy Forge's group, scanning electron micrographs that show the guinea pig organ of Corti after exposure to gentamicin, injections of gentamicin systemically. And what Andy found is within a week or so, outer hair cells were lost from the epithelium. And then a couple weeks later, there was evidence of inner hair cell loss, and most of the outer hair cells had died. And then several weeks later, 8 to 12 weeks, these supporting cells had expanded and formed this large supporting cell scar that had been described before as well, and no new hair cells were present. In the same study, Andy Forge also demonstrated, however, that there is evidence of vestibular hair cell replacement in the vestibular organs. So in the guinea pig utricle, this gentamicin treatment causes a swath of hair cells to be killed along the center of the utricle in the region called the striola. And if we look at high magnification in this area, we can see in the normal conditions that there are lots of hair cells there. At 2 weeks after gentamicin, however, there was extensive hair cell loss, and you can see some debris. And then within 4 weeks, Andy noted these small bundles had reformed in the utricle, in the striola region. And over the next few weeks, these cells acquired a more mature phenotype, indicating that they were likely regenerated. So we were interested in moving this work into the mouse model so that we could do genetic studies on it. And I worked with Ed Rubel and Richard Palmeter at the University of Washington to develop a mouse model to allow us to do selective and inducible ablation of hair cells in adult mice in vivo. And this work from Justin Golub characterized the vestibular response. And this mouse, which is called the PAL4F3 DTR mouse, which I'll call the DTR mouse, has the human diphtheria toxin gene injected or inserted into exon 1 of the PAL4F3 gene. Now, PAL4F3 is a transcription factor that's highly expressed in hair cells and not many other cells in the inner ear. And so what they reasoned is if this DTR were inserted into hair cells and hair cells expressed it, then when diphtheria toxin or DT was injected, then just the hair cells should be killed and all of the hair cells should be killed, sparing supporting cells and other cells in the inner ear. So to test this, Justin looked at adult 6 to 9 week old mice that were either wild type for the DTR locus or heterozygous. He gave mice 2 injections of the diphtheria toxin and then analyzed the utricles and the horizontal cristae at different periods after the diphtheria toxin injection, up to about 5 months. And what he found was similar to what Andy showed, which is that many hair cells are replaced in adult mouse utricles. Indeed, also Yoash Raphael's group had shown this for gentamicin damage. And this is illustrated here. And I should mention Yoash's work was in mice too. That in wild type mice, we see no damage as we would predict. We're now looking down at the surface of the utricle labeled for myosin 7A antibodies, and each one of these dots is a hair cell. And the normal hair cells are there. However, in the diphtheria toxin receptor mice, DTR mice, at 14 days post-DT, we saw extensive loss of hair cells, and hair cells gradually regenerated over time to fill in the sensory epithelium. And if we look at this quantitatively, our wild type normal hair cells, they're about, utricles have about 3,700 hair cells in them. 14 days afterwards, after DT, these numbers are reduced to 7%. And then over time, gradually the numbers increase to about 22% of control. And that's about 800 hair cells. This regeneration also happens in the adult mouse horizontal crista, as illustrated here. Here's a control crista labeled with antibodies to myosin 7A. This is DTR at 14 days post-DT, where there's extensive loss, and then there's addition of cells over time. Regeneration in the horizontal crista is not as robust as in the utricle. Only 15% of the hair cells seem to be replaced. How could we know that hair cells are really regenerated? Well, I worked with Brandon Cox to explore this with Stephanie Bucks, a postdoc in my lab running the project. And what we did is we found out that supporting cells that are labeled with a fluorescent marker before damage turned into hair cells after damage. We used mice here that allow us to label supporting cells with tomato, TD tomato, which is shown in magenta here, in supporting cells in adult mice, and we do this simply by giving an injection of tamoxifen. So here's the low mag view of the utricle with the hair cell marker in green and the tomato in magenta. And here's a higher magnification view where you can see the supporting cells in between the myosin 7a positive hair cells are labeled. Then we gave mice diphtheria toxin and induced hair cell death. And at 3 weeks post-damage, we could see evidence of hair cell regeneration. And when we looked at these cells at a higher magnification, we could tell that many of them had this tomato label that was present in only supporting cells before, signifying that these hair cells had come from supporting cells. But interestingly, others, and as well as our group, have found that all regenerated hair cells in the mouse utricle, indeed the mammalian utricle, are formed by direct transdifferentiation. We have found no evidence for supporting cell mitosis. Subsequent studies showed that the regenerated hair cells in the mouse utricle are type 2-like. And this, we looked with lots of different approaches, including different markers and transmission electron microscopy. And I'm showing you here the afferent nerve endings, which are different in type 1 and type 2 hair cells. So type 1 hair cells are invested by this calyx terminal, which are labeled here with neurofilament. It's labeled. And this extends around the whole cell body of the type 1 hair cell. Type 2 hair cells have bouton terminals that are small and extend along the cell surface. And we found that the vast majority of the hair cells that were regenerated do not have a calyx terminal. And here's an example of one type 1 hair cell that presumably survived that does still have a calyx. And when we quantified these hair cell numbers, we found that all regenerated hair cells are type 2-like. This is the control numbers of type 1 hair cells in green and type 2 hair cells in blue as measured per the number of hair cells per field. So this is before damage. After damage, shown below in the DTR mice, both type 1 and type 2, shown in black and red, went down to 9% of controls, but over time, the type 1 hair cells increased until their numbers reached about 44% of the normal levels here. Whereas the type 1 hair cells, those numbers just decreased over time. So this indicated that half of the type 2 population was regenerated, but no type 1s were regenerated. Now, this level of regeneration does not appear to restore vestibular function. And this is measured in a couple different ways. We've noticed that vestibular motor abnormalities arise after hair cell damage, but they persist after regeneration. And these include an unsteady gait, circling, and head bobbing. And with Jim Phillips, we also looked at the vestibular ocular reflex. This reflex allows us to stabilize eye movements during head movements. And in particular, the horizontal VOR allows us to stabilize our eye movements as our head moves in the horizontal plane. And it is a representation of function of our horizontal crista. So what we did is we rotated mice in the horizontal plane and looked at their eye movements and measured the VOR gain, which is a measure of eye velocity relative to head velocity. And if we look, we did this at different frequencies of movement or speeds of movement. And we looked in undamaged mice as well as mice that were DTR mice at different times after damage. And in all of the undamaged mice, you can see that the VOR gains are between 2.5 and 4. Right after damage at 14 days post-DT, there were no VORs elicited, no eye movements elicited when we rotated the animals. And at most of these frequencies, we did not see any recovery of that response. And we only saw very poor responses at 1 Hertz, which was the fastest rotation. So this indicated that the VOR was not restored as a result of type 2 hair cell regeneration. So this raises the question of whether vestibular function fails to return in mice because regenerated type 2 hair cells fail to establish mature features. So I had been working with Remy Pujol and Ruth Ann Etok and Tony Gonzalez Garrido in her lab to address this. And we've discovered that regenerated hair cells acquire hair bundles that resemble normal bundles in a lot of ways. So these are TEMs from Remy, and this is a wild type 2 hair cell. You can see the staircase pattern of its small bundle. After DT treatment, there's fusion of the bundle, and then these hair cells are lost. Then we see the reemergence of small bundles that have multi sort of small stereocilia at 28 days post-DT, and then at 170 days post-DT, these many cells go on to acquire this mature staircase appearance. With further analysis, we discovered that stereocilia height and diameter were within normal range, that the bundle orientations were also normal, and that the otoconal membrane and otoconia were intact. And Tony then went on to look at mechanotransduction by using a stiff probe to stimulate these small bundles in the regenerated type 2 hair cells while measuring their currents. And she used various stimulus frequencies here. And what she found in this particular mouse, which is 144 days post-DT, and in several ages, that there were mechanotransduction currents that were elicited by this stimulation, by the stereocilia displacement, and that they followed the stimulus and increased in amplitude as would be expected as the frequency increased. However, most of these regenerated hair cells had pretty small mechanotransduction currents relative to their normal counterparts. So this was one sign of potential immaturity. We also looked at ribbon synapses, and Remy found that there are ribbon synapses in these hair cells. And so the presynaptic ribbon is at the base of the hair cell and is a dense, structure surrounded by these vesicles of neurotransmitter. And the postsynaptic element is the afferent Bhutan ending in the type 2 hair cell. And what he sees here is just that in a regenerated hair cells at 140 days post-DT, this presynaptic ribbon with the vesicles and this postsynaptic Bhutan with this postsynaptic density. We wanted to quantify this, and so we used antibodies to the ribbon protein CTBB2 and antibodies to the postsynaptic glutamate receptors, GluA2, and we looked at and quantified the ribbons along the base of the type 2 hair cells here. And that's shown here. What you typically expect to see is a very close apposition of the CTBB2 in red and the GluA2 in green, which is a mature synapse along these processes. And work by undergraduates demonstrated that the percent of CTBB2 particles that co-localized with GluA2 was the same in regenerated hair cells as it was in our controls. So we looked at 2 wild-type controls, one with no DT and one with at 170 days post-DT, and we looked at DTR mice at 170 days post-DT. And so these hair cells seem to have the mature ribbon synapses that are, but as reflected by the co-localization of CTBB2 with GluA2. However, Tony also found some other signs in type two hair cells that are regenerated that may be another signal of immaturity. In voltage clamp, she noticed that these regenerated hair cells have two voltage-gated sodium currents that are typically uncommon in mature hair cells. So they're very common in immature hair cells, but as the hair cells mature, they're typically lost. And when she looked at these currents at multiple times post-DT, she found that many hair cells lost these currents, and they're shown in gray, but several of the regenerated hair cells had retained these currents even out to 300 to 400 days after DT. So this may be a sign that some of these regenerated hair cells are actually immature. And also, based on work from Christiane and Chabert's lab, it may also point to some immaturity in the afferent nerve endings, because when they deafferented nerves, deafferented hair cells, they found that they also increased their sodium conductances or sodium currents. So given that we had seen some signs of immaturity in these regenerated hair cells and the failure to restore vestibular functions, we wanted to ask if new hair cells can transduce a signal that's relayed to the brainstem. And we don't do afferent nerve recordings in my lab. And so what we did in collaboration with Jim Phillips is to look at stimulus-evoked CFOS expression in second-order vestibular neurons or vestibular neurons in the vestibular nucleus. And CFOS is a transcription factor that is a reflection of a change in neuronal activity when its expression is changed. So to look at CFOS, we subjected wild type and DTR mice to sinusoidal off-axis rotation. We placed mice in a beaker and rotated them at different speeds for 10 minutes. And this, we believe, this stimulus will stimulate all vestibular organs. Then we perfused mice with aldehydes 45 minutes later and sectioned the brain and looked in the brainstem at CFOS immunolabeling. And this is work that was done by a resident, a medical student, and a research scientist in my lab in collaboration with Jim and his trainee, or his another colleague in his lab, Leo Ling. So in undamaged mice, we found that CFOS expression was evoked in vestibular nucleus neurons by off-axis rotation. So this is a good marker of vestibular nucleus changes in activity. This is a negative control with no rotation, and we saw very little CFOS immunoreactivity in the medial vestibular and spinal vestibular nuclei. But after this rotation, off-axis rotation, we noticed lots of upregulation of CFOS in nuclei across these organs. And when we looked more closely at these nuclei, we found that they were located within neurons, as shown by this co-labeling with beta-3 tubulin, a marker of neurons. And interestingly, not all neurons upregulated CFOS in response to this stimulus. When we quantified this, we found that wild type mice with no rotation, as I mentioned, have very low levels of CFOS numbers. But these three different wild type controls all responded to rotation and with an increase in CFOS expression in the medial vestibular nucleus, shown here. And these are wild type without any DT, and then wild type animals plus DT at different times afterwards. So when we looked at animals that had just regenerated type 2 hair cells present, so DTR mice at different times, we found that they were not able to transduce and evoke changes in the brainstem nuclei. They were not able to evoke CFOS there. At 14 days post-DT, we expected not to see a response, but we thought if these regenerated hair cells were driving activity in the vestibular nucleus, that we would see an increase in CFOS here, and we did not. So one of the things that occurred to us is maybe this lack of rotation-evoked CFOS in the vestibular nuclei could be due to poor vestibular nerve conductance. Maybe the loss of hair cells or the DT itself could have injured the nerve and prevented the nerve from transmitting the signal from the hair cells. We don't think this is the case because we found that electrical stimulation of the vestibular nerve was able to evoke CFOS and vestibular nuclei even in the regenerated animals. We used a nerve stimulation protocol from Gay Holstein, published in this paper. We placed electrodes at the mastoids binaurally and administered 2 milliamps of sinusoidal current at 0.025 hertz for 3-minute intervals for a total of 30 minutes. And then 60 minutes after the session, we perfused with aldehydes and looked for CFOS expression. And what we found is whether it was a wild type plus DT or a DTR mouse a year after DT, this stimulation of the nerve was able to evoke responses in the nucleus, vestibular nuclei, suggesting that the nerve itself is, and the vestibular nuclei, neurons are functioning normally. And it's really the peripheral end organs that are not mature, not functioning properly. So to summarize vestibular hair cell regeneration research in adult mice from our group, so it appears the vestibular system is fortunately configured to allow some level of recovery. A subpopulation, about 44% of type 2 hair cells is regenerated naturally in rodents, but no type 1 hair cells are formed. No new type 1 hair cells are formed. Many regenerated type 2 hair cells appear to mature and become innervated, although they have some signs of immaturity. And I just want to note here that even human utricles seem to regenerate some hair cells. And this is work from Ruth Taylor and Andy Forge. However, the natural level of regeneration that happens in the adult mice is not sufficient to restore the normal vestibular motor activities or the VOR or even stimulus-evoked CFOS in the brainstem neurons. And so this raises a question of what's missing, what is needed. And one possibility is we're just not regenerating, or the animals are not regenerating enough hair cells. And this is something Alan Cheng's group has been looking into to see if expression of ATO1 in vivo, overexpression, can help. And what they found is this does indeed increase the number of hair cells that are regenerated, but it doesn't yield many new type 1 hair cells. Most of them are type 2 hair cells. But they did find some functional benefit as measured by VSEPs working with Sherry Jones, although it was variable across the animals. So maybe we need to promote maturity in the regenerated hair cells. I think this is a good idea, and I'm not sure how to do it, so I'm welcome to ideas, suggestions. Or maybe the type 2 hair cell population, even though 50% of it is replaced, is not enough to drive the neural activity. We need also some regeneration of type 1s. And my lab is working with Brandon Cox, Neil Siegel, Ruth Ann Etok, and Remy Pujol, and Jim Phillips to see if we can delete SOX2 in regenerated type 2 hair cells in vivo and see if this causes them to convert into type 1 hair cells. And we have exciting preliminary data, I don't have time to share today, that this does happen. We're checking to see if there's a functional benefit, and we don't know at this point. So I want to spend the next few minutes just giving a little overview on cochlear hair cell regeneration. And like I said, this isn't my field, and there are a lot of people working on it, and I'll try to summarize this. As I mentioned earlier from Andy Forge's work, unlike the adult vestibular organs, there are no hair cells regenerated in adult cochlea. Here are the hair cells labeled with myosin 7A and the normal vestibular epithelium and the organ of Corti. At 2 weeks after damage, we see loss of all the hair cells, most of the hair cells anyway. At 6 weeks after damage, the vestibular hair cells have regenerated, but the organ of Corti hair cells have not. Interestingly and encouragingly, neonatal cochlea of mice and other species, other rodents, has a capacity to form a small number of new hair cells naturally after damage. So this is work from Brandon Cox and Alan Chang, and what they showed was about 120 new hair cells are naturally regenerated from supporting cells after DT or ototoxic damage in the mouse cochlea. And other groups have demonstrated that overexpression of ATO or notch inhibition also increases the number of new hair cells that are formed in the neonatal cochlea. But in the mature cochlea, these pretty simple treatments fail to significantly augment regeneration or auditory function. Even though some new hair cells may be produced, there are not many of them, and they don't lead to great gains of function. So several groups are using different strategies to regenerate cochlear hair cells in mature animals. And what they're trying to do is learn why the adult mammalian supporting cells do not respond to signals that promote regeneration in neonatal mice, such as ATO overexpression and notch inhibition. And they're focusing on trying to reprogram supporting cells. And what this means is they are trying to manipulate the epigenetic landscape of these cells, the level of DNA methylation or histone acetylation, which may allow them to unlock the mechanisms that prevent conversion in hair cells, or prevent supporting cells from turning into hair cells. And there are lots of groups working on this here, and some of the approaches that they're taking are to overexpress the following in combination with ATO: transcriptional cofactors that may help ATO's function in the mature supporting cells, or chromatin remodeling agents such as histone deacetylase inhibitors or transcription factors that promote chromatin remodeling such as GATA3 and MYC. And if this fails, then it's possible that cell transplantation or introduction of cells from outside the ear into the damaged ear may be the approach that we need to take. And there is promising work from lots of different groups, particularly Ari Hishino and Carl Kohler's work, demonstrating that multipotent stem cells can differentiate into hair cells when the correct factors in three-dimensional environment are present. And these organoid studies are a very exciting new direction of hair cell regeneration research. So I want to end there and thank the members of the Stone Lab who contributed to the work that I presented, the trainees, my lab members who've been with me a long time and whose work I appreciate enormously, undergraduates who've worked in the lab, and my other contributors, collaborators, Jim Phillips with Leo Ling, Brandon Cox, Ruth Ann Etok with Tony Gonzalez-Garrido, Remy Pujol, Neil Siegel, Ed Rubel, and Richard Palmeter, and Gay Holstein, and I want to recognize my funding sources. So thank you all for listening, and I'd love to entertain questions.
Very good. I guess we don't have a method for audience applause yet on Zoom. I hope they're working on that. But in the meantime, there are a couple questions in the box, and there's probably time for a few more if anybody wants to submit them. First question has to do with regeneration in the avian ear, and was wondering about the kinetics, or I assume the timing between the formation of a new hair cell and the time at which one sees functional recovery. How long does it take to reestablish function from the time after a hair cell regenerates?
Yeah, that's a great question. In the chick BP, regeneration happens over a pretty discrete period compared to what I showed, for instance, for the mouse utricle. So we can see cells beginning to re-enter the cell cycle between 18 and 24 hours. We can see evidence of early differentiating cells by 3 to 4 days. And then we can see evidence of bundles by 6 days after damage. Really don't see recovery of ABR thresholds to normal. It's demonstrated by work from, for instance, Diane Durham's group and Doug Gerard, until 3 weeks after ototoxicity. And so this whole population of hair cells are, they're being formed in over the first week, and then they're differentiating So I'd say it takes about 2 weeks for a hair cell to, grow its bundle, become reinnervated, synapses to mature, the overlying tectorial membrane to regenerate, et cetera, and then to have restoration of the thresholds. Behavior, like auditory recognition of calls and song learning, for instance, which Sarah Woolley's with Ed Rubel shown return, take longer, up to a month probably, or a little longer than that. And interestingly, vestibular hair cell regeneration can take several months, and work from Ed Rubel and David Dickman's groups, your work as well, have shown that reinnervation, particularly of those type 1 hair cells with the calyx, can take months. So similarly, restoration of function can take a long time in the vestibular epithelium.
Great. I have another question about regeneration in the mammalian ear, and this, the question has to do with the survival of the afferents after hair cell loss, and was wondering whether degeneration of those afferents might be one of the reasons why you don't see a more complete restoration of vestibular function after regeneration.
Yes, wish I had more time to talk about that. Remy and I have been looking at that a bit in the mouse utricle, and we've been looking at that with transmission electron microscopy and also with confocal microscopy, looking at different nerve markers. And what we are seeing is that the vestibular afferents, we've mainly looked at afferents, we really haven't looked at efferents a lot, retract down to the basal lamina, and for some period of time, maybe between 7 days and 21 days, there really aren't a lot of nerve elements in the epithelium as these hair cells are being regenerated. And then by 21 to 28 days, we can see growing neurites re-emerging in the epithelium, and synapses are forming already by 3 to 4 weeks on these regenerated hair cells. We've looked at numbers of ganglion neurons. Capstone student in my lab, Lindsay Schoblum, has done that, and now an undergraduate student's following up on that, Hannah Zaini. And we don't see any loss of vestibular ganglion neurons, even out to 6 months after damage. And we've also looked at the vestibular nucleus neurons, and they also don't show any signs of degeneration. So it could be that the neurites, grow back in and establish synapses and look pretty normal, but they really aren't normal. I mean, after all, most afferent neurons receive inputs from type 2 and type 1 hair cells, and they're only able to reconnect with the type 2 hair cells. And so they're getting different responses and different signals than normally. So there's still more work to be done, but we don't think there's any permanent loss of vestibular ganglion neurons after hair cell damage and regeneration.
Another very related question asks why you don't see any calyx nerve terminals on these regenerated hair cells.
Well, that's a $1 million question. And I think, I doubt very much that this has anything to do with the challenge of forming a new calyx. I think this has to do with the lack of a type 1-like cell differentiating. And I'm pretty confident when I say that the type 1 hair cell differentiates in the absence of a calyx. Okay, so it's responding to different cues. Why we don't get type 1 hair cell differentiation in the, among those regenerated cells is unclear. But what's interesting is that type 1 hair cell production is limited to the embryonic period. So work from Alan Chang and Joe Burns and Mark and I showed that no type 1 seem to be formed after birth, and but half the type 2s are. And work from Stephanie Bucks and Brandon Cox and I showed that even type 2s are they undergo turnover over the life of the animal, and they are regenerated. So type 2s show a lot of plasticity, and type 1s don't. They can't be formed after birth, apparently. So there's some strong mechanisms inhibiting that, and we'd like to know what they are. But we know from our preliminary work where we're able to cause type 2 hair cells to convert into type 1-like cells, that when they acquire those type 1-like properties, which may be, downregulating markers of type 2s, upregulating markers of type 1s, they attract calyces to them. So if we can regenerate them, the calyces will come. Even in a mature animal, there's a lot of neural plasticity. The nerves are happy to adapt if you put a type 1 hair cell there. So I think we need to figure out if supporting cell progenitors that can form new type 1 hair cells are restricted to the embryonic period, or if there's signals that otherwise could drive a bipotent type 1, type 2 hair cell progenitor to form one or the other, whether those are lost in the postnatal period.
Yeah, there've been a lot of questions coming in on this general topic, and we probably won't have time to get to all of the specific ones, but I think you've covered that area pretty well. Another area that seems to be attracting a lot of questioning has to do with transdifferentiation, supporting cells to hair cells. And so what do we know about the very early events that occur when a supporting cell is making that decision, if you will, to transdifferentiate into a hair cell?
Yes, that's a very important question. And the answer is, all we know is sort of what we've observed about the timing and a few changes that happen in different genes that are expressed there, et cetera. In our work, we work from John Cafaro in the 2007 showed that it seems that supporting cells begin to transdifferentiate into hair cells in the chick BP, okay, even before the hair cells are lost. So it looks like something's changing in the hair cells, whether it's loss of activity or loss of spontaneous neurotransmitter release, or something's changing in the hair cells that causes ATO1 to be upregulated in supporting cells before hair cells are even lost. So it looks like transdifferentiation is initiated first. We don't really see, and Doug Kotanch and others have not seen a lot of supporting cell division until day 3 after damage. And at that point, there's been almost complete hair cell loss. So it seems like the signals that are driving cell division are happening later. And Dave Roberson had an interesting idea working with Doug Kotanch that maybe it's the loss of the supporting cells as they transdifferentiate into hair cells that initiate supporting cells to divide, and replacing those supporting cells that transdifferentiated. And then maybe notch signaling regulates or causes some of those precursor post-mitotic cells to become hair cells too. Work from Mike Lovett and Mark Warshall on the transcriptomics of early transcriptional changes are happening in the BP, may also reveal some of the signaling pathways that are different at the early stages versus the later stages. Stefan Heller's working on this as well. But we don't really know. It seems, for instance, loss of notch inhibition will either push post-mitotic cells toward the hair cell fate, or supporting cells toward the hair cell fate. I hope that answers the question.
Yeah, another category that's getting a lot of attention has to do with the function of these regenerated type 2s, and you showed some data on that, but do we really know that they are transmitting a signal to the vestibular afferents on the one hand, and do we really know that their stereocilia, kinocilium, are really embedded in the otolithic membrane on the other hand? how do you account for the lack of really much functional recovery, despite a good deal of recovery of hair cell numbers, at least among the type 2s?
Well, I think that's a complicated question. I don't know the answer to it. I can tell you that Tony in Ruth Ann's lab tried really hard to patch on to these boutons post-synaptic to the regenerating hair cells and see if she could evoke responses when she displaced the bundles. And they're really small and they're really hard to patch onto. And we weren't able to answer that question in that study, unfortunately. We see signs that these cells should be functional. We see signs that they seem immature. Like I mentioned, we aren't able to, in my lab yet, to do the afferent nerve recordings, for instance, while we rotate the animal and see if crista afferents respond, for instance. That's certainly something we'd like to do, and Jim and I have talked a lot about doing that. We don't know if the conocilia of the type 2 hair cells is embedded in the otoconal membrane. We've been also trying to do that. We know the otoconal membrane and otoconia are there. We know that the hair cell bundles are oriented appropriately and that they have these eccentric conocilia. So the bundles look the right way, but we haven't been able to see the conocilium projecting into the otoconal membrane. And remember that the type 2 hair cell stereocilia are so short, they don't reach the membrane. So that we're looking for that. That's another clue, I agree, that we'd like to know the answer to.
Someone was curious about the use of organoids, presumably iPSC-derived organoids for studying hair cell development.
Well, yeah, I mean, I think the organoids are going to prove to be very helpful across a whole range of studies, looking at ototoxicity, looking at agents that might promote regeneration of different cell types in the organoids. We've known for a while that embryonic stem cells can form hair cell-like cells, but it wasn't until they grew in the 3 dimensions that we really saw how beautifully they can differentiate in mammals. I think one thing that's striking about these hair cells that form spontaneously in the organoids, I shouldn't say spontaneously because there are a lot of factors added and stuff too, but they tend to have a vestibular phenotype, and they have features of vestibular cells. And so I know a lot of people are working on trying to get them to acquire a more auditory-like phenotype. I think the question remains, what are you going to do once you get these differentiated cells? How are you going to get them into the ear, right? You can't take a slab of tissue and inject it into the cochlea. I don't know how to do it. So I think that poses a problem for transplantation. And it might be that people get clever and develop sensory organs elsewhere in the ear, or approach the vestibular regeneration challenge that way first, because the architecture is less exquisite and challenging. But I think I'm really excited about the organoid literature, and I'm sure it will explode a lot in the next 10 years.
Recovery you see is, exclusively type 2 hair cells. In the normal case, what is the contribution of type 2 hair cells to the different physiological measurements that you're making to assess function? Do you think that you're measuring, vestibular evoked responses that in the normal case are mostly due to type 1s, and that might be a reason why you're not seeing any improvement when you have only type 2s recovering in the epithelium?
Yeah, I think it's interesting how regeneration studies can shed some light on the function, normal function of things, and raise new questions. No, we don't know what the function of type 2 hair cells and type 1 hair cells are, really. And as I mentioned, most of the afferent neurons, particularly in the periphery of these organs, receive input from both type 1s and type 2s. And so they're computing inputs from both of these cell types. And it's generally thought, based on work from different groups, that some of the things that we're studying, like the VOR and the VSEP, and other people are studying, may be primarily driven by hair cells in specific regions of the utricle. For instance, the VSEP is most likely driven by fast neurons, fast hair cells, type 1 hair cells that are located in the streola, and the VOR is likely driven by cells in the periphery. But we don't know specifically what the type 1 and type 2 hair cells are doing in those contexts. It is interesting in a study that from Ed Rubel's group, that the VOR developed late in chickens that were regenerating, and it correlated with the regeneration of type 1 hair cells temporally. So that provides a little bit of additional evidence that maybe type 1s are important in the VOR response, particularly at higher frequencies. So I hope we can test the hypothesis that type 1 hair cells are useful to vestibular recovery by converting these regenerated type 2 hair cells into type 1 hair cells. But we don't know if they're going to be really type 1-like enough. And we've also talked about deleting subpopulations, or one particular type of the hair cell versus the other, using genetic approaches and seeing how that impacts these different behaviors. But I think that the possibility exists that type 2 hair cells do something we don't even know how to test, okay? And so it could be that they're different types of physical features, like the short bundles and relatively large surface areas, may make them better suited to respond to different types of stimuli. And it might be that we're simply not looking at recovery of those stimuli appropriately with our testing measures. And I think it's a recent paper from Elizabeth Glowatsky and Suresh Sadeghi raised some interesting questions about type 1 and type 2s, because they show that efferent inputs from the brainstem to the utricle, mouse utricle, have different effects on the calyx versus the type 2 hair cell. So they activate calyx activity, but quiet or hyperpolarized type 2 hair cells, so that they have less inputs. And so this would suggest that modulating the inputs of type 2 hair cells and allowing type 1s to be the only hair cell speaking, so to speak, in certain circumstances might benefit the animal. And so I think that's a clue that these hair cells do different things.
Another thing that's come up among the questions here, and I think I can state it pretty simply, and that is any speculation as to why the mammalian vestibular organs have at least some capacity for replacement of hair cells via transdifferentiation, but the cochlea lacks that ability?
Yeah, a lot of hand waving, probably. There's a lot of conservation in the shape, cell type, structure of the vestibular organs relative to the hearing organs across the non-mammals and the mammals. And so, it's tempting to speculate that relative similarity allows mammals to have some vestige of the capacity to regenerate hair cells. It's also interesting that type 2 hair cells resemble hair cells in amniotes more than the type 1 hair cells do. And so that the type 2 hair cells are the only hair cells present in these non-mammalian species, some of them that regenerate like fish and amphibians, are, you know, they only have type 2 hair cells. So it could be that that could be one possibility why type 2 regeneration also is more likely to happen, because it's a more phylogenetically old cell type. Another interesting speculation is that in some animals, like songbirds and fish that use vocalizations, there is seasonal plasticity that occurs, and they have changes in the central nervous system and the peripheral nervous system that, for instance, Ali Coffin showed it in midshipman fish. There's addition of hair cells in the saccule and death of them as well seasonally. And so it could be that having this plasticity be able to increase and decrease hair cell number over the season has provided some sort of you know, survival benefit to species that communicate with their vestibular organs. All right. And then finally, you know, hearing has required us to requires an organ with fixed mass and stiffness, and so one way to really throw that off is to have supporting cells dividing and adding new hair cells. And so it would seem potentially that, you know, any mutations that lead to more cell division, more formation of hair cells in the cochlea would not be advantageous and selected for. And so that may be one way in which the regenerative capacity in the cochlea has been lost, or just never developed in mammals.
Okay, maybe I'll add one more question, and then maybe we can wrap things up, unless you want to continue. That has to do with supporting cells. When you have an appreciable amount of transdifferentiation occurring in the regenerating utricles, say, that means you're losing supporting cells. Does that have any, do you think that has any impact on the sensory organ? And if so, are they replaced?
Yeah, we haven't looked at that on a sort of week by week basis. What we've done is looked at the end of regeneration, like 5 months after DT, how many supporting cells are present. And even though there've been, you know, 800 new hair cells regenerated, we don't see a lot of supporting cell depletion. There's a little bit, but not much. And so it seems that there is some pretty crafty way in which mammals replace supporting cells. And it may be cell division. I mean, it could be that all of us who've tried to see dividing cells just didn't capture them. And I remember you published a study where you saw dividing cells in human utricles and also in guinea pig utricles. So it could be that they're there and the divisions are happening infrequently enough that we are missing them. Or it could be that supporting cells come from another source, they're replenished from another source. I mean, one attractive hypothesis is that they may come from the cells that surround the sensory epithelium, the transitional epithelial cells, which are fairly undifferentiated and which express a lot of supporting cell markers normally, but there's no definitive evidence for that. But anyway, I would not say that the small degree of supporting cell loss that's happening is impacting function or functional recovery in any way.
Okay, I think this is a good time to wrap it up. I'll point out to all of you whose questions were not answered, I tried to consolidate, you know, some of the questions into one general topic. But the questions that were not directly answered, these will be forwarded to Dr. Stone, who will then get back to you via email with an answer. So I think we've reached the end of this session. Thanks a lot for a great talk, and I want to thank all the audience members for attending. And this is turning out to be a really nice format and a mechanism to communicate among ARO members. Thanks, everyone.
It's really fun. Mark, thank you so much for your participation in this. And I wish I could look out into my camera and see you all. I think you're there. Miss you and hope you all stay happy and safe.
Great, you too.
Thank you.
Bye.
Bye.